Welcome back to Bio 111. This is chapter 14. We're going to be looking at DNA structure and function. All right, the first thing we're looking at is the central dogma of DNA, or central dogma of biology. I've mentioned this a couple of times in this class. The flow of genetic information is from DNA to RNA to proteins. The DNA is found in the nucleus, as we've seen. It contains all the information needed to make the proteins, and from there needed to make all the things that proteins might make. Remember, the proteins are a generic term for molecules such as enzymes. And enzymes are the machinery that go in and catalyze and produce other products, such as DNA itself, ATP, remember we talked about ATPase, and many other things. All right, but the central dogma, you have DNA, which is storing all the information. For DNA to be used, we have to transcribe it into RNA. DNA and RNA are very similar molecules, and so that's why we call it transcription. We're just changing it, we're just writing it in a different notation. So we're taking DNA and we're having some machinery in the cell transcribe it into RNA. And then that RNA is translated into proteins, right? We're going from a completely different language. If you remember, DNA and RNA are made up of what are called nucleotides, adenine, thymine, cytosine, guanine, and uracil. Proteins are made up of amino acids. So we're going from having an alphabet of those A, T, C, T, or A, T, G, C, U, to having an alphabet of amino acids. So we have a translation there. All right. Main idea here is that the flow of information is going from DNA to RNA to protein. All right. All right, so DNA has its roots. The discovery of DNA has its roots in 1869, when scientist Frederick Miescher isolated phosphate-rich acetic acid from compounds found in the nuclei of leukocytes, white blood cells, in pus from hospital bandages. He must have been very curious to be going after that sample. He termed the material nuclein, since he was able to isolate it from the prepared nuclei. Eventually, this material became known as DNA, or deoxyribose nucleic acid. Now, later on, Frederick Griffith discovered that when he exposed mice to two different strains of a bacteria, that he found two different results. So he had the R strain, which was non-pathogenic, meaning it did not cause disease, and the S strain, which was pathogenic and caused disease. In fact, it killed the mice that he injected. Well, he decided to determine what would happen if he killed the S strain, the strain that actually caused the death, and inject that along with the live R strain into a mouse. When he did that, the mouse died. Now, this is part of a larger experiment. We're just cutting out a large part of the experiment, but we're looking at the part that's important to us. Even with a dead S strain, the live R strain survived. Well, that's interesting. Now, when he looked at the mouse, the dead mouse, he was able to recover a live S strain bacteria. All right, so we killed the S strain. We injected only dead S bacteria and only had living R bacteria when we injected it, but we recovered live S strain bacteria. So something happened. Somehow, information that made these bacteria R or S was transferred from the dead bacteria to the living bacteria. So Griffiths concluded that something had passed from the heat-killed S strain to the R strain, and thus transforming the R strain into the S strain in the process. All right? So we know at this point that there is something that is capable of moving between the bacteria. Now, still didn't know what that was, what was being carried between these two strains of bacteria. This group of scientists we see right here, they decided to determine what that material was. And so what they did is they took extracts from the S strain of the dead mice, and they systematically degraded specific macromolecules, proteins, DNA, and injected these into mice with R strain. They found that when they did not degrade the DNA, the R strain would transform or would turn into the S strain. And they therefore determined that DNA was the molecule that was carrying this information. Now, that gave a little bit of credence to the idea that DNA might be this hereditary particle that we saw in the previous chapter that Mendel described, that we already have assigned at least visually to chromosomes, which we don't know what chromosomes are quite yet. But DNA was a very simple molecule as far as these scientists understood. DNA was far simpler than it was expected was needed in order to transfer all this information. We've got a lot of information needed in order to make living organisms, bacteria, much less humans. And they thought that more likely these complex proteins that they had discovered were more likely responsible for transforming this data. Of course, we know today that it's DNA actually holds the data to make the proteins. They didn't know this. And so they decided, well, let's do an experiment and figure out what is actually moving between what is being used to, can we rule out protein? So they looked at a biological activity where they labeled the proteins of a bacteriophage. Now, a bacteriophage is a virus that will infect bacteria. They labeled the protein with a radioactive phosphorus, and they labeled the DNA with radioactive phosphorus. They labeled the protein with a radioactive sulfur. There's sulfur in some proteins, sulfur in some amino acids. There's phosphate in all there's phosphorus in all DNA. All right, so what they did was they waited for this bacteriophage to inject the DNA, or inject something, we didn't know it was DNA, inject something into the bacteria. They knew the bacteriophage was injecting something because it had the ability to transform these bacteria. They waited for that to happen. They, using a blender, shook the bacteriophage off of the bacteria, and they put it into a centrifuge. Now, a centrifuge is a device that you spin your solution, and the heavier parts go to the bottom of your test tubes that you're spinning, and the lighter things don't go quite as far down. Well, the bacteria are really heavy, so they form what we call a pellet at the bottom of the tube, and the virus, the bacteriophages, they stayed at the top. All right. Now, what they found was that the bacteria now had the radioactive phosphate in them, thus indicating that it was DNA that was being injected into these bacteria. And so this gave more information, or gave more evidence to the idea that it was DNA that was acting as the transfer of genetic information. Now, another scientist by the name of Chargaff found that you could break down these nucleic acids, DNA, RNA, into pieces, right? These are the nucleotides, right? And he found there were 4 nucleotides that build up DNA: adenine, thymine, cytosine, and guanine. He also found that you had roughly the same amount of these in organisms of the same, well, samples from the same individual, samples from the same species, but when you went to different species, they would come in different amounts, the 4 nucleotides, right? Now, we know today that the information in DNA is stored in the sequence of these nucleotides. So it makes sense that different species might have different amounts of each of these nucleotides. But was something that was kind of unexpected for him was that some of these, particularly, they were grouped together such that the percentage of adenine equaled the percentage of thymine, and the percentage of cytosine equaled the percentage of guanine. Now, a bit perplexing. At this point, we did not know the structure of DNA. We're about to see the structure of DNA, and this rule is going to make sense once we see that structure. All right. So that's the end of section 1. We'll look at section 2 in the next video.